Hello everyone and welcome to GenQA's Focus On webinar for this month, whereby we're very delighted to present Professor Dame Lynn Chitty, who has very kindly agreed to come today and tell us about national implementation of a rapid fetal sequencing service in a public health setting, and give us an idea of the benefits and the challenges that the laboratory has faced for the service. Now, we're also joined today by my colleague, Dr. Dave Grugin, who is one of the deputy directors at GenQA, who is also going to tell us about some of the related external quality assessment schemes that are provided by GenQA for the fetal sequencing and other related diagnostic services. So I'm Professor Sandy Deans and I'm Director of GenQA, and as I say, we have great pleasure in welcoming Professor Dame Lynn Chitty today. Lynn is Professor of Genetics and Fetal Medicine at Great Ormond Street Hospital and is UCLH Gosh part of Institute of Child Health, and has been really fundamental in the development of the fetal sequencing service across NHS England. We'll also have an opportunity at the end of the presentations for you to submit some questions, and we'll have a live question and answer session. So I would encourage you to pop any questions you'd like to ask either Lynn or Dave or myself around GenQA into the questions box that hopefully you can see on your screen now. So please think about those questions and put them in, and I'm now going to hand over to Lynn to ask you to please give us your presentation. Thank you.
Okay, thank you very much, Sandy. So as Sandy says, I'm going to talk about how we implemented rapid fetal sequencing in our public health service. Most of you know that the NHS National Genomic Medicine Service was reconfigured in 2018, and our genetics service is now delivered from 7 genomic laboratory hubs, and rapid fetal sequencing is delivered from 2, from Central and South in Birmingham and North Thames at Great Ormond Street. And this is what the service does, basically. So NHS rapid fetal sequencing, or we call it R21, that's the number it's given in our National Genomic Test Directory, began in October 2020. It is for the indications are fetal structural abnormalities detected on prenatal imaging, where multidisciplinary team review, including clinical genetics, considers a monogenetic disorder is likely, and diagnosis may influence pregnancy or early neonatal management. And I stress, and I will continue to stress throughout this talk, that this is now a multidisciplinary, it's a team sport. It's no longer send off a test and get a result back. Indications specifically are indicated here, so major anomalies in multiple systems, suspected fetal dysplasias, large echogenic kidneys, etc. And over time, we have added more indications, including high drops, agenesis of the corpus callosum, and small for gestational age, with no evidence of placental insufficiency. This is the pathway. So we do fetal and parental trio whole exome sequencing, and the analysis of that sequencing is done using a panel of around 1,400 genes that we know may cause fetal or neonatal abnormalities. And that panel is updated regularly, so we review new genes every 6 months and add to it. So the pathway here is that imaging detects fetal abnormalities. There is a discussion locally between genetics and fetal medicine, or genetics and the obstetric scanners. If they decide that this is likely to be genetic, they discuss it with the referring, with their relevant genomic laboratory hub, who approve it. And then samples are collected, DNA extracted, and QC is sent to the testing genomic laboratory hub. Before any sequencing is started, QF-PCR is done to exclude aneuploidy, and microarrays are done in parallel for speed. So if it's accepted, there's trio WES is done in the genomic laboratory hub, and with the necessary bioinformatics and variant interpretation. And this is discussed again with the referring team, so MDT again. The time, a target time for a preliminary result is 2 weeks, and then 3 weeks from receipt of samples for the final report, because we often need to confirm the variant and discuss and issue the report to the referring team. And the only things that we report are pathogenic or likely pathogenic variants related to the phenotype or indication for exome sequencing. So we do not report other pathogenic variants unless they have particularly relevance to the parents or their reproductive future. I highlight the MDTs again, as I said I would do, because it really is very important that we discuss this across the team. So the service has evolved. It's evolved, as I've explained already, to increase the eligibility criteria. We update the fetal panel regularly. And we've also established a clinical oversight group to provide expert advice in cases where there is uncertainty on eligibility. And this group is made up of representatives from all 7 genomic lab hubs who have expert clinical knowledge and experience in fetal anomalies with a monogenic cause. So where are we? Well, these are the data from the end of September this year. We had just over 2,500 inquiries, and of those we accepted just over 2,000. The reasons for declining cases were either because they did not fulfill eligibility criteria, alternative tests had been suggested, or that the parents had decided to terminate the pregnancy, or fetal demise is imminent, or parents declined invasive testing. Of the 2,000 that we've accepted, we have subsequently reported on, it must be now 1,600 because this is a couple of months ago, and we have 494 diagnostic variants. Many of these cases did not proceed, as you see, around 500 did not proceed. Again, many for the reasons I've already explained. Many declined invasive testing or declined exome sequencing, fetal demise delivered, or there were laboratory issues with insufficient DNA or maternal cell contamination, or the diagnostic results came through from arrays or another test done in parallel. I think it's important to note that those cases which were either declined or did not proceed are still eligible for sequencing after birth, and we call that R412 or whole genome sequencing. Our diagnostic yield is about 31%, and the mean turnaround time to final result is 14 days. And you can see that over the years, as we've been going now for 3 years, the number of referrals and tests done has increased. And the heat map there shows you that we are getting cases from across England, and this service is only for England, it's not for Wales and Scotland. We have a very wide range of genes have been reported, and the heterogeneity here supports the use of a gene agnostic or very large panel approach. We've also detected a number of rare variants, which highlights the importance of data sharing. Some work that we did early on, it's not quite finished yet, looking at clinical impact. We have shown that exome sequencing impacts on management in 56% of cases. More cases where there's a diagnosis, but nonetheless, over a third of cases which remain undiagnosed, the fact that we've got a diagnosis, the exome sequencing has been done, has helped make decisions. And there's some examples there. So either involving the relevant team at delivery, or identifying a very small baby as having Russell Silver, allowing the pregnancy to continue to term rather than induce early for growth retardation, or in the last case here, a diagnosis of Holt Oram enabled expert counselling by the parents and gave the parents the confidence to continue the pregnancy. I think it's important that we look at the inheritance patterns here, and around 60% of the inheritance patterns are autosomal dominant. And the important thing here of the dominant ones is they're not all de novo new dominants, but a pathogenic variant was inherited from an unaffected or mildly affected parent in up to 10% of cases. So we cannot rely on inheritance filtering alone. And so parents need to be aware that in doing this test, we may well find that they themselves have a condition which they've passed on to their child. And that can be quite challenging in terms of counselling. There are a number of laboratory challenges encountered, and I'm not going to go through them in detail, although I recognise this is a laboratory meeting. I am not a laboratory person, I'm a clinician, so I'm going to focus more on the clinical aspects. But there are challenges in variant interpretation, incidental findings, which you see in fetus and parents. And because we're doing trios, we can find things in parents, mosaicism, copy number variants. Some of them are too small to be detected on microarray, and we're picking things up on exome sequencing that we haven't picked up on array. Non-paternity, mosaicism, and then of course diagnosis not detected by fetal exome sequencing include genes which are not on the fetal anomalies panel, which means there has to be careful pre-test counselling. And I think one of our really big issues is incomplete or poor phenotyping. Remember that we are dependent on prenatal imaging, so we can't see whether the fetus is blind, deaf, has intellectual disability, and we are really, really dependent on our expert sonographers to give us as much detail as possible. So that's what the service is and some of the issues. And what I really want to do now is talk to you about the Express study, And this was a study funded by the NIHR, HS&DR to evaluate the new prenatal exome sequencing service to provide evidence to improve the quality of care and equity of access for parents having prenatal tests. Now, this is a study which started on the 1st of October. It wasn't meant to start on the 1st of October 2020, in the middle of COVID. It was meant to start earlier, as indeed was the sequencing service. So the research we've done here is slightly compromised by COVID, but nonetheless, I think we've got a lot of really useful information. The study itself comprised 6 work packages, which are detailed here. We did a lot of interviews with health professionals and with parents. We looked at outcomes to look at the diagnostic yield and referral rates. We identified new pathways. We looked at the ethical issues. We've done a health economic analysis, which was accepted for publication yesterday. And the PhD Foundation are in the process of putting together all of our data to write a report on this for policymakers and stakeholders. So we've talked about the laboratory issues. We've talked a little bit about the non-paternity and the ethical issues. We've talked about the challenges for trio sequencing, and I'm going to give you some examples. The need for updating the fetal panel, because we learn more about genomics all the time. The scale of this task is quite enormous, and the number of people that were required, and there are very few people, few of us volunteering at the moment. But we are reviewing 300 or 400 genes every 6 months. The eligibility, how do we get a robust evidence-based approach to extending eligibility? And what about the phenotyping? We have identified that there are very variable standards across the country. And furthermore, we know that fetal phenotypes can evolve, and there may be new phenotypes, completely new phenotypes of things we don't see postnatally, or new phenotypes which present of conditions which we know well postnatally but present differently in the fetus. And there's very variable clinical care pathways across the country, and there's a need to identify optimal pathways to do some standardization. So I'm going to give you some examples. So we thought we would be dealing with 7 genomic laboratory hubs and 7 pathways, but we were very mistaken here. And basically, we identified the fact that we have 17 regional genomic services that all vary in approach to implementation. The approach, they have different staff involved at different points, different approach to multidisciplinary team working, very variable access to expert prenatal genetic counselling, and a very variable standard of phenotyping. And this is sort of the main 7 or the main 6 structures that we identified. So some of these services are led by clinical genetics, some are led by clinical genetics and a genetic counsellor, and some are much more fetal medicine and genetic counselling. So it's very variable, and we've interviewed people across the whole country and looked at these pathways. And these are some of the, this is not a great slide, but some of the things that clinicians wanted. They wanted both clinical genetics and fetal medicine to devolve the service to fetal medicine. But this is going to take a lot of education, and education for clinicians was recognised as being a key key point required. Many of them wanted broader eligibility criteria to improve access to this testing, but how do we do that? They definitely wanted a systems change, more administrative support and more staff. There's quite a lot of time taken in counselling patients here, and it's quite a burden, a clinical burden on units. And they wanted ways of sharing knowledge and improving communication. And we have indeed a national education meeting now. So let me just highlight for some of you, and I recognise that you're laboratory people, so I apologise for all of the ultrasound slides here. This fetus was referred with prenatal edema, which you can see here, and a nuchal translucency. Now, no structural abnormalities detected, normal scan otherwise, so this was not eligible. But the testing GLH requested the images for review, and when they saw it, they thought, well, this baby not only has prefrontal edema, but micrognathia, and it looks like Cornelia de Lange, so expert review thought this looked like Cornelia de Lange syndrome, so we accepted it for sequencing, and it was de Lange. So this case would not have been sequenced without expert review of the ultrasound images, and this is a classic example of why we need experts. These conditions are rare, and most people in district general hospitals will not see these cases. They'll see the occasional one, but they won't get a lot of wealth of experience you can get in tertiary referral units. Another example, a baby with short, long bones, small chest, mild micrognathia, difficult scan. We asked that the GLH requested a tertiary fetal medicine review, but this was denied. And the preliminary result was a collagen mutation, which is compatible with osteogenesis imperfecta. But because that's a very specific and very non-specific finding, and there was no other signs of OI, no bowing, no nothing, a review of the phenotype was requested. We finally did get the images sent to us, and you can see here, these are very bent limb long bones. Both of them are bent. And if you look at the growth chart, this is absolutely classic of osteogenesis imperfecta type 3. So this variant from likely pathogenic was upgraded to class 5, and this was the result confirming it. Again, we need expert phenotyping, and it can be difficult. So this is a case again with a small chest here. This is a sagittal section of small chest, short long bones and a narrow chest. In this case, we had two potential diagnoses. One was Jones syndrome with a DYNCH2H1 variant, clearly pathogenic variant inherited from dad, and a variant of uncertain significance inherited from mum. This is a recessive condition. So these findings are compatible with that. But there was also a de novo unclassifiable collagen variant, which could cause achondrogenesis type 2. So this is dominant, so very different. So here the parents decided to terminate the pregnancy, and the radiograph was compatible with Jones, which is recessive. So we were able to upgrade the maternal variant and give a diagnosis. So messages here, again, pathology can be very useful, and we need to document any new fetal phenotype genotype associations. The other thing is that if we do a sequencing and don't find a variant, we need to keep scanning. So this is an example of a case with a cardiac abnormality and a normal microarray. We got this CAT6A deletion, which was classed as class 5, but it didn't fit the phenotype. So it was reported as a potential class 5, but without fitting the phenotype. But they continued scanning this baby, and at 31 weeks gestation, the classical signs of craniosynostosis, which is well reported in this postnatally, appeared in the fetus. So now the variant was found to explain the phenotype. So this is another case here where phenotypes may evolve prenatally and allow reclassification. And parents need to be aware of that, and we need to keep an eye on these pregnancies. A classic example of this is increased nuchal translucency. So this is a review of 100 cases of isolated raised nuchal translucency. That's that thickening at the back of the neck. Many of you will know about this. Some of you will have had scans to look for this. And if you look at the overall, the diagnostic variants, there were 11 diagnostic variants in this case of 100 cases of isolated nuchal translucency. But if you looked in more detail, if those cases where it remained isolated, the 75 cases, there were only 2 diagnostic variants. And it's the cases where they developed additional features that were picked up later in pregnancy had a very high diagnostic hit rate. So evolution in pregnancy is really important. I've talked to you about new fetal phenotypes, and this is a classic example. Here, this is a baby with fetal hydrops. That means extra fluid all around the lungs and in the abdomen here. It had a big D2 variant, and this was a pathogenic variant. But the trouble here is that this has not been previously associated with hydrops, although other neuromuscular conditions have been presented with hydrops. So it's a class 3 VUS. And what this is really associated with are muscle problems in the lower extremities. The parents terminated the pregnancy because of the severe ultrasound findings. And the following week, we identified a paper published linking this BIC to CD to hydrops. And the parents had consented to post-mortem, and the myopathic changes were consistent. So this variant was upgraded to likely pathogenic. So the message here is phenotypes may evolve. They may be very different from those that we recognize postnatally. Post-mortem can still remain important to further define new phenotypes, and it's very important to monitor the literature and share new information. Another example here, again, this is absent cerebellar vermis, a TMEM67 compound, heterozygous was identified, pathogenic variant from dad, and a hot variant from mum. We couldn't upgrade this variant based on the current evidence, but there was a maternal variant of uncertain significance reported as likely pathogenic on ClinVar. So what we did was we contacted that lab, and detailed information from that lab allowed us to upgrade that from a variant of uncertain significance to likely pathogenic, again confirming the diagnosis. And actually, I would encourage all of you, if you come across cases like this, to try. There are a lot of efforts ongoing to improve the fetal phenotype correlations, including some of the things listed here, but publishing them so that we can help other people understand new phenotypes and new phenotype genotypes is really, really important, and can be quite quick, as in these short genotype phenotype reports in prenatal diagnosis. The other thing is that we need to think about the family history. You know, I said that 10% of the dominant conditions were paternally inherited, in many occasions from an unsuspecting parent. So this is a case here, again of hydrops. You can see the pleural effusion here and the thickening at the back of the neck. And the preliminary R21 result was an EPHB4 maternally inherited variant, type grade 3, a hot variant. When these cause capillary arterial malformation syndrome and hydrops fetalis, so very compatible with the fetal phenotype, but it came from mum, and we didn't know anything about mum. So we discussed this at MDT and decided that what we really needed to do was examine mum and take a detailed family history looking for features of this variant. And lo and behold, mum, grandma, and a lot of other members of the family had signs. So swollen legs, cherry angiomas, lymphedema, and they all had the particular variant. So we were able to upgrade the variant to likely pathogenic. This also changed the pregnancy management, because hydrops is one thing, but this particular variant could cause AV malformation. So we needed to do an echo and an MRI to make sure the patient had no AVMs. So the message here is that actually, as part of this process, we need to take a detailed family history and parental examination. And this is challenging because most referrals come from fetal medicine. So we need to embed genomics more in fetal medicine. We need to train our fetal medicine doctors to think genomically, or increase the collaborations between genomics and fetal medicine. And don't forget, occasionally, in about 5% of cases, you may get 2 diagnoses. And in this particular case, had we just looked at the de novo variants and not looked any further, we would have identified this PTP-N11 de novo heterozygous variant, which can cause Noonan's. This is dominant, but looking in more detail, we identified an EPPH maternally inherited heterozygous variant, which causes the condition that we just described. So again, examination of mum here found the cherry red angiomas and a history of edema, and detailed family history and parental examination again must be required, and we need more education, as most referrals come from fetal medicine. The 2 main ethical issues that we identified were around equity of access, and equity of access in many respects actually, issues around the eligibility criteria, because this limits access, and how best do you define eligibility, laboratory analytical processes, because we're confining it to a panel of genes, and the awareness and education of clinicians, and some women are not being referred or referred very late because of lack of awareness. And then the geography, variable access to expert phenotyping, scans, parental travel costs, etc., etc. The other ethical issue was one that I don't think we can do very much about, is timeliness and its impact on parental decision making, largely focused around the law around termination of pregnancy, where in England, the law states that you can terminate a pregnancy for any reason up until 24 weeks gestation, but thereafter you have to have significant risk of substantial handicap. And the decision making in the absence of prenatal sequencing can be quite difficult. And the other thing that we also found was that the importance, and I stress the word importance, of prenatal sequencing results is being overemphasized. All of these fetuses are being sequenced because they have fetal abnormalities, and those abnormalities carry prognosis in their own right, and I think that we are sometimes ignoring that. So how should we define eligibility criteria? And I think this is quite challenging. So the Page study and the study from US, which basically sequenced any fetus with any abnormality without any genetic input, showed that there are certain specific groups which have a higher instance of monogenic conditions, so skeletal abnormalities, hydrops, and multisystem abnormalities. But then the overall year was about 10%. And if you look at that, so this is the Lord study, pathogenic variants, any structural abnormality, 11%. Petrosky, the US study, again, 10%. But if you look at cases which are selected after clinical genetic review and where they're thought to be a likely genetic monogenic etiology, you can see that the diagnostic rate goes up to about 32%. So I think that one of the things we can say is that pre-screening by clinical genetics will increase diagnostic yield, and that will be a good way to define some eligibility. How else should we be defining this? Should we be using systematic reviews? And there are a glut of them out there. Well, the problem with systematic reviews, looking at the eligibility criteria, is that on the whole, there is poor, if any, definition of ultrasound standards. We don't know whether the abnormalities are isolated or associated with other abnormalities. Little or no information on other tests done. And just referring to one of the systematic reviews that I'm involved in, so I'm able to be critical of this without offending anybody apart from me. This is a systematic review on isolated long bone shortening with no evidence of fetal growth restriction. The systematic review came out with a figure of 48% of these cases are monogenic. But if you look at the online appendix, more than 2/3 of these conditions these cases had features which were detectable on ultrasound. Thanatophoric dysplasia, osteogenesis imperfecta, etc., etc., etc. So the standard of ultrasound here has been pretty awful. I need to come just briefly to the experience of parents, and this is summarising an awful lot of work. But we have interviewed 48 parents, offered prenatal sequencing, and integrated the findings of the health professional interviews, and these have been published. Unfortunately, these came from cases mostly from parents who accepted prenatal exome sequencing, and there was a wide range of experience with results, but we do need more diversity. And this is one slide which sums it all up. Patients were overwhelmingly positive about the service, incredibly grateful to have it. It helped with decision making. It helped people make an informed decision, and many of them were very positive about the support throughout the testing process. Others, however, felt that there's still uncertainty across testing. You know, we haven't done the entire genome. There could still be something there. What if we don't get an answer from this? What does it all mean? Some parents felt that they needed more support. The timing of the results was highlighted, and we've talked about the 24-week legal termination law. And many parents wanted more information than we had time to give, so we need to think about that. And the other thing that was very pertinent was that follow-up is important regardless of the results that you're getting. Whether you get a positive result or whether there's no finding, you need to follow this up. You know, this quote here, we have no explanation, we may never. That will always be at the back of our minds. Suggestions to improve care included clear information about what genes to be tested and what could be reported, multiple opportunities for questions, information in different language and different formats, provision of emotional support alongside information giving. And I think this is a role for fetal medicine midwives and genetic counsellors, and continuity of care here, and the need for follow-up whether a diagnosis is made or not. We have addressed one of the issues and co-designed a prenatal sequencing animation with our PPIE group. This is now translated into 12 different languages and is formally under review at the moment. There are other things we need to consider. So should we be doing whole exomes, whole genomes, panel testing? What is the role of long-read sequencing? What should we be reporting? Likely pathogenic and pathogenic variants in all genes, only those presenting in the prenatal period, genes that are amenable to early therapy for the baby. How are we going to decide what we should report? And importantly, should we be looking for additional findings in the fetus, the parents? Should we be looking for the same or different ones? And when and how should we be doing it? I'm pleased to say that some of these issues will be addressed in the Prenatal Genomic Network of Excellence, And at North Thames at Great Ormond Street, we're going live with whole genome sequencing in January, but we will still be using the panel test prenatally to deliver R21. But we will be exploring whether or not we should look for findings and what else we should be doing. So what do we need to do? Well, we need to build on the work already done. We need to ensure expert phenotyping of all fetuses, including pathology where appropriate, ensure expert prenatal genetic review. This is definitely multidisciplinary team working, and we do need a lot more education for all health professionals and parents. We need to improve the counselling and develop information in multiple formats, and we need to ensure expert post-test counselling and support regardless of result. We need to resist the over-reliance on sequencing results, remembering that most of us are sequencing abnormal fetuses, and we definitely need to share our data and publication. How might we do this? Well, I think that we need to identify and support organisations who may not have had previous experience, as these teams may need additional support. We need close collaboration and communication within and across services, facilitated through a range of strategies, including joint appointments, MDT meetings, regular communication, etc. If existing collaborations are not in place, there is a need to bring different services together to develop these relationships. We definitely need more education for all disciplines, and there is a need for increased staffing across these models. We need to streamline our logistics and processes, and we need to think about how we expand the national guidance for prenatal exome sequencing. And some examples include, for example, outlining the importance of adapting local pathways to fit local context with expectations and clarity around which part of the pathway should not vary and which parts may be better adapted to locally. Some processes need further clarity. We definitely need to improve access to expert phenotyping. We need to think about developing MDTs nationally and improve IT and explore the use of virtual MDTs. And in the Genomic Network of Excellence, we are already doing this to a certain extent. We have established a small working group which includes fetal medicine, clinical genetics, NHS England, patient advocate, perinatal pathology and a commissioner. We've reviewed the data and suggestions include the development of a national network of MDTs building on existing infrastructure with a virtual element so all cases can access the core requirements, which should be expert fetal phenotyping, expert perinatal genetic expertise. We should be able to use these MDTs to triage ongoing pregnancies for R21 and also triage cases with pregnancy loss, stillbirth and termination pregnancy for genomic testing and or pathology, because we are very short of perinatal pathologies. We recognise that this will take time and require investment in consultant time, education for all, and more perinatal geneticists in particular. So finally, I think we've come a long way with fetal sequencing, but we have a long way still to go. And thanks to the many patients, many organisations that have helped with all of this work, and the funding you can see there has come from a variety of sources over the years. And finally, for those of you who are interested, we have now got a number of papers published from the Express work, which are listed here. So thank you very much for your attention, and I'm happy to take questions if there's time.
Thank you, Lynn, very much indeed. What a fantastic piece of work. It just shows what we can do on a national basis when there's infrastructure and expertise there. So thank you for that. We're going to take some questions. There are questions coming in, so I've got quite a few for you, but we're going to do a live Q&A after Dave has gone through his presentation around the associated external quality assessment schemes within the fetal sequencing space. So Dave, over to you for your presentation, please.
Thank you. I think Emma's just going to put some slides up for us.
Wonderful, thank you.
Oh, and she has.
Wonderful, thanks, Dave.
Move on a couple, Emma. That's great. So I'm just going to talk about a couple of EQA schemes that we offer that I think are relevant to the audience that we've got today. And starting with our trio sequencing EQA. Currently, we've got two versions of this EQA. We've got a prenatal version and postnatal version, which is anything that's not prenatal. But just to go back a bit, this is developed over quite a few years now. So we started this EQA just as exome sequencing in 2021. At the time, it was just data interpretation, so we provide some results. It wasn't DNA samples, and they were just SNV results. We've moved on over the years. 2022, we expanded that and included CNVs in that as well. Again, it was data interpretation only. 2023, we offered an optional DNA-based trio as well as the data interpretation trio. And that actually went really well. We had a really good uptake of that. So this year, 2024, we decided to split the EQA into prenatal and postnatal versions. There's one trio in each for this year. The assessment's actually ongoing at the moment, so I can't present anything about that. But next year, we're going to expand again. So for the prenatal trio sequencing, so we've changed the name slightly. So we've taken the word exome out. So if somebody did want to do whole genome sequencing, that would be possible. It's just trio sequencing. And we're going to offer a data interpretation trio and a DNA-based trio. And the data interpretation trio, as well as we'll be able to offer more interesting scenarios that way, probably. But also it opens it up to labs that don't do sequencing themselves in-house. So hopefully they'll be able to participate as well. Moving on to the next slide. This is just an example, just in case you wanted to know the kind of data that we do provide. We have to provide it far enough along the pathway that everybody can participate. So we could provide examples if you want to know if you could participate. Obviously, the DNA-based test, you just test and report in your normal format. So going on to the next slide. So this is a pilot EQA that we started offering last year. Again, this is currently open for 2024, but in 2023, we offered a carrier screening EQA. DNA samples from a couple, labs could test and report them in their normal manner. So it might be that they reported the individual separately, or it might be that they test them together, depending on the strategy they have in their country or their lab. We also had quite an extensive survey that we sent out with that EQA, asking about testing strategy, methodology, customers, that kind of thing. And you can see an example of the report that we issued last year at the bottom. There's quite a lot of information there for labs. I think it's quite useful for people for benchmarking. We've done a similar system this year, but instead of one couple, there's two couples. So we've expanded it a bit, hoping to cover the testing strategy of more labs. Again, there's another survey, so that's ongoing. But if you wanted to enroll for that, the enrollment for 2025 is now open. That'll be a similar situation to 2024, two couples. Going on to the next slide. And as Lynn was talking about, MDT is very important nowadays, and we offer an EQA for MDT. We started this last year. It was a pilot. It's a pilot again this year. I'm talking about rare disease here. We also offer an EQA for molecular tumor board, but this one's rare disease. So for 2023, we provided a clinical scenario with some genomic testing results that we hoped would require an MDT. And then we asked for centres to provide their MDT preparation, so probably the variant classification and what they needed to take to the MDT, and that could be in whatever format. So it could be something formal, like a specific form, or it could just be an email conversation, a phone conversation, whatever you normally do in your centre. And then we asked for a record of the outcome of the MDT and the discussion that took place, and then also the clinical report. For 2024, we've offered two cases. So there's one prenatal and there's one postnatal. So you could pick and choose which one you wanted to do. That EQA is actually currently open now, but it doesn't close until the end of January. So if anyone did want to join in now, that would be possible, or enrollments are also open for next year. And we'd really encourage people to join in for this, because it's really good to get in at the pilot stage, and we ask for your feedback on how you found the EQA, if you found it was useful, that kind of thing. So really encourage participation in that. And that's me done. Back to our question and answers.
Great, thank you very much, Dave. That's really interesting. Lots of flavour of the whole end-to-end process into the EQAs that's been delivered. Thank you. So we've got a couple of questions. Lynn, I'm going to start with you. You mentioned about clinical genetics review helps to increase your diagnostic yield. So who else currently is able to actually request fetal sequencing? Is it only clinical genetics?
Yeah, it's only clinical genetics.
So is there any plans to expand that out to, fetal medicine units or other areas of the
if I had my way, yes, I'd embed this in fetal medicine, but they've got to be trained. And I think that's
very true.
But so we actually have just appointed, he started last week, a fetal medicine genomic fellow. So he's an obstetrician, and we're going to train him in variant interpretation. And he's done some work at UCH looking at the funny fetuses, and then he needs to know the genomic bit of it. I mean,
Okay.
I'm trained in, you know, looking at dysmorphic children as well as fetuses, and we need more people who can look at the dysmorphic fetus.
Yeah, yeah, yeah. It's quite a niche skill set, isn't it? That
You're
actually
beginning-
follows on really nicely to one of the other questions that have come in, which is how do we move forward with working with fetal medicine and increasing their genomic knowledge? You said that that was one of the blockers. How
well,
do
I
we
think
do that then?
we start at medical school. We definitely start with obstetric training. I mean, it's not obstetricians and gynecologists. In fact, all general clinicians need a better genomic knowledge. There isn't enough training. They're training. They should be trained. You might have a maternal medicine fellow. You should have a genomics fellow. So I think we need to get the deaneries and the training sorted out. There's something else that came into my mind. But yeah, the other thing which I did when I was at UCH was I embedded a genetic counsellor in the unit. So we had a genetic counsellor who was funded half by UCLH and half by Great Ormond Street, where the clinical genetics service was, and she worked across both services. Absolutely invaluable, both in terms of educating the midwives and the staff, but also managing the cases. So
absolutely, and I think, and we're really privileged in the UK to have so many genetic counsellors. I know we're short of them, but it, you know, compared to other countries where they do not have genetic counsellors, I think
they need to be embedded in the fetal medicine unit. They need to actually go and sit there and see patients there and work there. And we need to be encouraging that.
Brilliant, thank you. Dave, one question's for you come in about your carrier screening EQA. So which genes do you cover? Because I guess we know that everybody does things slightly differently depending on which countries the test is being performed. So which ones do you cover? And I guess how do we choose which ones we cover?
It's actually really difficult to, like, I mean, this is why we have pilot EQAs, I guess, because we have to work out what we need to offer to people and what people are offering in their lab as well. So hence the kind of extensive survey that we carried out, which is really useful, obviously, for coming up with this year's EQA. The answer is really that we ask for people to carry out just their standard carrier screening tests that they would normally do in their lab. We can do some benchmarking between labs. Obviously, we have an expected result for the sample, and we have validations done, but they might not necessarily cover all of the genes. So there's a bit of benchmarking between participant results as well. Obviously, it makes choosing the sample difficult because we have to have consent to test for basically anything on these samples, which is fine for the samples that we use. But it's very much sort of information gathering as well at this stage of the pilot.
Yeah, thanks, Dave. I guess it's an example of how challenging EQA delivery can be. It's not just saying, oh, well, we'll set up this test EQA and find samples and run it out. We have to really scope out what's happening globally.
Yeah, and get feedback from participants about what we've done and what they'd like us to do for the future as well. Yeah.
Yeah, great, thank you. Lynn, back to you. Do you postnatally reanalyze data if the pregnancy, for example, is continued?
Not routinely, no. If there's a clinical indication, if there's a new finding postnatally, then maybe yes. We will be doing this as part of a research project. We're getting ethics approval at the moment. When we go live with whole genome sequencing, we'll be doing reanalysis after birth, but to see what the increased yield is, etc., etc. But we do not do this routinely clinically. You can't do it.
Interesting question, though, isn't it, about if the evidence is there to actually do that?
Well, we hopefully will provide some evidence, and there may be particular circumstances where reanalysis is appropriate.
Sure. And final one for you, Lynn, the fetal panel. So is there, you said that even though you're going to be doing whole genome sequencing from the whole exome sequencing, is there any benefit of also doing a gene agnostic approach, even though you're just applying currently the fetal gene panel?
I don't think so.
I'm putting you on the spot. That's quite a difficult question, isn't
it?
it?
Question, but I think the answer is probably not, because we're already looking at the parents, and if you do gene agnostic, we'll do it after birth, but we're not doing it before birth. I think you'll find things that you don't necessarily want to find.
Yeah. So I guess it's fetal sequencing is very different service than a postnatal service.
I think it is, and I think that's something I'd really like to stress, Sandy, actually, because I know there are people that want to, for example, incidental findings. You know, these incidental findings may not be incidental. in the prenatal setting, because of the evolution of phenotypes, the change in the new phenotypes. And I think we need to be really clear that what we're doing in the fetus is different from the neonate. It doesn't mean to say you don't need to look at it carefully and have genetic input, but I really think, I know some people want to sort of merge the whole thing and think it's all the same. It's not the same. It's very different, and the choices are different.
Yeah, very important point. Thank you, Lynn. And Dave, finally, one last question for you is about your MDT EQA. So how does MDT EQA work? As all the EQAs that we usually perform are lab-based, so how is it slightly different? Which is a good question.
Yes. Obviously, in this case, we're providing a scenario, we're providing some results. It's going to depend on the year as to what we offer. We try and come up with interesting scenarios. And actually, also, if people have interesting scenarios that they think that we could include, we'd really encourage you to get in touch as well, because that can be really useful for this kind of situation. It's all based on the scenario. But yeah, we provide the clinical information, and we provide what the variant is. Obviously, we don't give you any classification, and quite often, that can be involved. It can be, classifying something that might come up as uncertain significance, and then you'd go through your process and try and come up with something for it, or maybe exclude it. It might not be relevant at all. So it's a scenario. It's not sample based at all, and it's for the whole centre as well. So the whole team. We don't want it to just be a lab based thing. We'd like the whole team to be involved.
Yeah, great. Thank you. It's a interesting one, actually. It's the way things are developing, isn't it? Thank you. Emma, can we go on to the next slide? I would just like to say to our participants, thank you very much for joining us today. We will ask you for some feedback. So if you don't mind, please do complete our survey, because we develop these focus on webinars in response to what you would like to hear about going forward. So it's really valuable to hear your views. In time, and I think it's about a month's time, the webinar will be available to view on the GenQA YouTube channel, and you'll also automatically be sent participation certificates for joining us today. Please sign up to a newsletter. You can hear all about when we're out and about at conferences or particular papers that are being published, or these focus on webinars, and lots of other news about registration going live. And just to flag, we're delighted to be joined by Ros Hastings on the 2nd of December, because many of you will know that the ISCN 2024 book has just been published just last week, and she is going to talk us through how the changes from the 2020 guidance book and about improving the nomenclature for cytogenomics. So I'm sure that will be incredibly useful for many of our participants. So finally, just a huge thank you to Lynn. Thank you, Lynn, very much for summarising the work, the amazing work that's going on for fetal sequencing, and also Dave for the related EQAs that are coming from GenQA. So thank you everyone for joining. Take care.
Thank you.